Okay, so now we're recording. So introductory session 1 at UCSF. Everyone should see a screen that looks similar to mine with a number of icons. Those icons are thumbnails for different data sets. So I want to explain a little bit about the view that you're looking at, because this is the view when you open Imaris that you will see first of all. So at the top left-hand corner of your screen, you'll see a number of icons. There are 3 that we're going to work with today. The first one is labeled Arena, the second one is labeled Surpass, and the third one is labeled Vantage. Those are different windows and different views within the Imaris workflow. So Arena is the first one you'll see when you launch the application, and this is basically where we do our data management and also batch processing. So the idea within the Arena view is that you organize your data based on your experiment or your samples or your organisms, whatever it is you want to do, and you can use what we call assays and groups to organize your data. So there's 2 little folders. They look like baskets. One is a round basket, one is a square basket. They're up on your top toolbar on the left-hand side, and those are what we use to organize your data within the Arena window. So an assay is equivalent to a master folder, and a group is the same as a subfolder. Okay, so maybe your assay, just as an example, would be if I click on the assay button, you'll see that it will come up and ask you to name it. So as an example, I'm going to call this one Lindsay. And if I click OK, you'll see a new assay appear in your list on the left-hand side with the name. If you are clicked on that assay or master folder, you can then click on the group and add in subfolders within that assay. So this is if you're in a lab and you want to separate out each of your work. You're in a shared situation and you want each of the users to have their own assay, and then within there you can have different experiments or different days or whatever it is. Okay, so I'm just going to put in here test, and you'll see that it will add now a subfolder into this Lindsay assay. Okay, to get data in, you're going to click on the location you want the data to go. So if you want it directly in your master folder or your assay, you click on that. If you want it inside the group, make sure you click on your group, and then go to the little red blob called image, again on your top toolbar. If you click on that image icon, it will open up the open dialog. Okay. Now in this dialog, I want to show you something. There is a pull-down list called Files of Type, and this pull-down list has a complete listing of all of the file formats that we support in Imaris. So we talked about this morning. It's important to have your files come in with metadata. So this is just a list of all the different files that we support. One thing I also want to mention, if you want to open up multiple files, select all of those files holding your shift key down, and that will select multiple, and it will open multiple files into that group or assay. Okay, now we already have an assay created, and it's called Imaris Demo Images, so you only have one in your configuration. Inside of that, the thumbnails that you see are different data sets that have been imported in for you already. Now to open one of those data sets, you're going to double click, and we're going to start with the one that is called Retina. Retina? I said that weird. Retina. So it's red and green. It's this one right here. If you can see my icon, this one right here. So if you go ahead and double click on that icon, it will open up that data set into the Surpass view. So you're no longer in Arena, you're now in Surpass. Okay? Now, we're going to use this data set. It's very basic, but we're going to use this data set to do a little bit with the visualization techniques that we have in Imaris. Before we do that, one last thing. If you do not want to use Arena, Arena is for people who want to organize their data, batch process, be able to see thumbnails. If you do not need it or want it, you can come straight into the Surpass view and go to the File menu and Open, or you can drag and drop your data directly into the Surpass view. Does that make sense? So Arena is an option. You do not have to use it. You can click on the Surpass tab to get straight into the Surpass view. This is where we're going to do all the visualization and all the analysis. If you want to open a file, you go to File Open. You get the same dialog we saw before, or you can drag and drop your file directly into this window. Okay. So first thing, you'll notice over on the left-hand side of the screen, you have a list. And those lists are what we call, that's a surface scene list, and it contains objects. Objects are items that we're going to be adding into your 3D data set. So when we start putting in surfaces and spots, you're going to see that we're going to add to that list. There are always 3 created automatically when you open a data set in Imaris. The first one is called a light source, second is a frame, and the third is a volume. We're going to use all 3 of these today, so you know what they are, but the volume object is the image data itself. Okay? You'll notice that each of them have a little checkbox next to them. That checkbox will allow you to turn them on and off. So if you do that with the volume or the frame, you'll see that that object will be hidden or shown. Okay? The frame is that white box that you see around the edge of the image. That is there to give you that perspective, that 3D perspective. It's also there for some people for the purpose of measurement. There's a grid pattern in there. People that are tracking like to have some kind of a reference. So if you don't like that frame, you can turn it on and off. Okay, especially for snapshots and movies, if you want to remove it, you can turn it off over on the left-hand side. One other thing, if you click on these different objects, you'll see that down in the bottom left-hand corner, there's a configuration for each of the objects. Okay, so in the frame, you can configure it to take the grid off, you can take the box off, you can make it look however you would prefer it to look. Okay, so basically each of the objects can be configured or modified based on that little configuration window. The volume object is the one that we're going to be working with next. So the volume object is the image data. Everyone okay? We're good. Okay, sorry. I thought it was not working. The volume object is your data, and this is also where all of the 3D rendering controls are. So remember this morning I talked about 4 different rendering techniques that we have in Imaris. The default is called a maximum intensity projection, and that's what you're all looking at right now. If you look at the configuration for the volume object, you'll see that you have a blend, a normal shading, and a shadow projection option. Okay? We're going to look at blend first of all, and I'm also going to ask you guys to do one more thing. There is a little window, and if you don't have it, I'm going to show you how to find it. And that window is called Display Adjustment, and it floats on top of the Imaris window. Does everyone have one? If for whatever reason you do not have one, if you go under the Edit menu, there's an option called Show Display Adjustment, and that will open it up if it's previously hidden. Okay? The display adjustment is really important for contrast enhancement and basically having control of the display of your channel information. So this display adjustment will show you each individual channel within that data set. It will be colored with a lookup table. That will depend on the metadata, incidentally, as to how it comes in or what color it has been applied with. And each of those channels has 3 black triangles on it. Okay? The lowest, or the one over towards the left-hand side, is your background control. The one furthest over on the right-hand side is your brightness control, and the one in the middle is your gamma control. So I'm going to switch back to the maximum intensity projection for a moment. So each of your channels has independent contrast enhancement tools for each of the channels that you have in the image. Okay? Also, each channel has a little checkbox, so you can turn the channels on and off independently. That can be very useful if you're trying to adjust the display for one channel. You can turn the other channels off while you do that. Okay? Now, I'm going to show you how you can change the color table, because a lot of times when you open up data in Imaris, the color table may not be exactly the way you want it. Maybe it looks orange when you want it to be red, or maybe you're red-green colorblind and you want it to be cyan and magenta. So I want to show you how you change the color table. So each of your channels should have a little name next to the checkbox. If you click on the name, it will open up a window like this that will show you a color wheel. And that color wheel allows you to choose the color you would like to apply to your data set. Okay? So you can choose whatever color you would like. And there's also one other option that I'm going to show you. Right now we're using what's called a base color, one color for the entire channel. We also have an option called mapped color. And this is for those people who want to apply a heat map or a custom color table to their data. A lot of people looking at intensity changes in an image may apply a heat map so they can see changes in intensity. So if you click on mapped color, you'll see that down at the bottom there is a little pull down list of some custom lookup tables. And just to show you one, there's one in there called Fire. And if you click on it, it will show you what that color table looks like. And basically this is showing you what the minimum intensity and what the maximum intensity would be and all the colors in between. So not something you're going to use every day, but for people looking at changes in intensity, this one can help you to visualize those changes. Okay, so I'm going to go back to base color, and I'm actually going to cancel because I'm perfectly happy with the colors I have here. Yes?
For the mapped color, is there a way to kind of output the color scale?
Output the color scale, you mean export the lookup table? Or
Yeah.
so you can export the lookup table. So in that little dialog, the mapped color, this little window that we're looking at here, actually allows you to customize your own lookup table. So there is an option to import and export a lookup table. So if there's any particular one that you want to use, you can actually use this lookup table in other applications as well. So you can export a lookup table out, or you can bring one in that you've already made. Is that answer?
What I mean was like, say if it had like a certain, say the darkest value.
You mean like a scale bar?
Yeah.
Okay, so no, there's currently no way to apply a scale bar for that. That would purely be just a visualization tool to look for differences and changes. However, if I can add that to our feature request list and see if that's something, if you would be interested in that. Okay? So, yes?
When you adjust the min, max, and gamma of each channel, is that just for visualization, or does that change how?
It's a great question. So the question was, if we change the display or the contrast enhancement using those little black triangles, does that change the underlying data? And the answer is no, it does not. It is a color lookup table. It's purely a display. So anything that you do with those little triangles to change the display is not changing the underlying intensity values in there. Okay? So I'm going to go ahead and cancel out of here and leave the color tables as they are, but I wanted to show you where you could change that. Now, the next thing we're going to do is, now that we know where the contrast enhancement tools are and the channel controls, we're going to switch into the blend mode. If you're not already there, click on the volume object, click on blend. And that's going to bring you into this mode where the vessel that we're looking at looks solid. And hopefully your background looks gray, not black like mine. I've set up my preferences a little different. So has everyone got an image that looks kind of like what I see? Okay. So I'm going to basically talk to you a little bit about this dialog over on the left-hand side that's underneath your rendering options. Okay? This is where we are going to control the opacity of the different channels. Okay, so remember, blend adds opacity and shading, so you have control of what intensities are made solid and which intensities are ignored. So over here on the left-hand side, you can see both your channels, and if you click on them, you will see the intensity display for each of those channels. You're also going to see this little plot on here, which indicates the intensity value. This minimum one is showing you the intensity values that are being ignored. Anything below that value will be black. And then we have this little slider bar here that we're going to use in a second, which will allow us to change opacity. So this axis here is your opacity, and this here is your intensity value. Okay? If I grab this right in the center here for the red channel, you'll see that if I pull it up and down, it will change the opacity of that particular channel. And depending on where you have that placed, it's basically applying it to a particular intensity range. So if you drag it down or towards the left-hand side, it's going to make more of the dimmer intensities look more solid. And as you bring it over to the right-hand side, it's going to focus more on the brighter structures. Okay? Now you'll notice I just pulled the whole slider down. which will allow you to kind of set a particular gradient. But for now, we want to just focus on the fact that up and down is your opacity, and then depending on the structures of interest, you can move it from left to right to get different intensity values. Okay? Now, these controls over here for opacity are separate from the display over here. So if it looks dark, you can use your display adjustment to bring up the brightness. Okay? So you can use the display adjustment palette as well as your blend controls together to optimize how you visualize the data. Now, as I said, this is now a blend reconstruction. So if you click on your 3D view and move it, you'll see that you'll be able to move and reorient that data from whatever direction you would like. Okay? If possible, I'd like you to orient it so that you're looking through the vessel, kind of like what I'm doing right here. Okay? And this is a great example of why blend rendering for 3D is so useful. Okay. Do you see, if you look inside the vessel, do you see those lumps and bumps inside? Okay. Try changing it to a maximum intensity projection again. Can you see those lumps and bumps? Okay. Can you see the surface structure? Kind of, but not really. So the blend technique gives you a completely different visualization and a different way to see three-dimensional structures. Okay? Now, there's one other thing I want to show you. Also, I was doing this all on the red channel. You have the same exact controls for opacity for your green channel also. So you can adjust those independently of one another. And the same with the display for that too. Okay? Now, the other thing I wanted to show you while we're in here is this new feature we have called Peak. I won't spend too much time on it, but I just want to give you an idea. So I'm going to turn my green channel off and I'm going to focus on the vessel. So we're going to look at the vessel, and down at the bottom of my blend controls there is an option called Graph Shape. One is called Step and one is called Peak. The default is called Step. That's what we're using right now. If you switch to, make sure you're clicked on the red one because that's the one we're going to work with. Click on the Peak option and you'll notice that you'll actually now have 3 points instead of 2. Now what this will allow you to do is you will have control of each of those points individually. You'll be able to spread them further apart and bring them closer together. And this middle point here is still controlling opacity. Okay? So this is basically isolating the intensity values that are inside this range that you're looking at that's described here in the min and max dialogues. You can then and hopefully see something similar to what my image looks like right now, which is basically a vessel that looks like it is hollow. You can also adjust the middle peak to adjust the opacity of that as well. Okay? So this is the blend, this new blend technique that allows you to render it so it looks hollow, so you can see other structures that are inside a little bit better. Okay? So I wanted to show you that one. This is one that I think that a lot of the people that I was working with yesterday, at least I saw, could benefit from using a technique like this. Okay? So the same can be done with the green channel, but for now I just wanted to show you where that function was. Okay, so I'm going to take Peak off and go back to Step because I have another function I want to show you, and that's another rendering mode, which is the one labeled Normal Shading. Okay, so we just looked at Maximum Intensity Projection, Blend. The third one is called Normal Shading. If I click on that, you're going to notice that the structure is going to look bright, and it's also going to look even more solid. Okay. So what we're doing right now is we're using a blend mode, but we've added in an additional light source. So over on our left-hand side, we had an object called a light source. I didn't describe it or explain it initially, but I'm going to use it now. If you click on that light source, you'll notice a little line will be attached to your image at some point. It's probably oriented in some direction. I'm going to now, with your mouse wheel, scroll in and out, and you'll see that that will control your zoom level. And as you zoom out, you're going to see this little light source icon or tool. Okay, it looks like a plunger. Everyone should hopefully see it. Now, you'll notice that if I'm dragging the image around, that light source stays in exactly the same location, okay? I can't access it, I can't move it, I can't turn it. So there are two modes in Imaris with the mouse. The first one is what we call navigate mode, which allows you to zoom and rotate the structure. The second one is called select mode, and this is allowing you to interact with structures in the 3D view. So if you, a shortcut for this, maybe I'll show you the original. So the control of this is over on the right-hand side under pointer. There's a select and navigate. You're going to find that you're going to switch between those two modes all day, every day when you use Imaris. So having to go over and click on something is kind of tedious. So there is a shortcut for it, and when you start using Imaris, you'll find that you'll have one hand on the mouse. You're going to have the other finger on the escape key, okay? The escape key is the shortcut between navigate and select. And as you tap the escape key, you'll notice that the icon on your screen will change from a pointer to a double arrow. The double arrow is the navigate, and the single arrow is select mode, okay? Now, when you're in select mode, now you can grab objects in that 3D view. So if you try grabbing your light source, you're going to notice that as you change that orientation, that it's going to, change the gradient of light. Okay? You can also put that light source behind your structure, which has a weird look. I wish it actually stopped you from doing that, but you can actually put it behind. So try and keep it in front of your structure so that you can actually see that light reflection. Okay, so I wanted to show you that because this is ultimately what gives you the ability to emphasize particular parts of that 3D structure if you have control of where the light is coming from. Okay, so one last thing. You'll also notice with the normal shading mode, you still have control of opacity, so you can combine that with your light source to really make it look exactly the way you want it to. Okay? Okay, the last mode. The last mode is called shadow projection. Now this one is by far the most computationally intense. So if you're working with data sets that are bigger than about maybe 1 to 2 gigabytes, this one is going to take time. Okay? And what you're going to notice is if we go to that shadow projection option, which is the fourth rendering control, you're probably going to notice a little progress bar that appears in the bottom right hand corner. Okay, so I'm right now in shadow projection. The orientation of light impacts this greatly. So if you were playing around with the light and it's behind the structure now, the front of your object will look black. Okay, so make sure your light source is kind of in front of your data set. As you make changes, though, you'll probably have already noticed that as you make changes, you may not see anything happen. We do that on purpose so that you don't have to see this constant rebuild of the structure. So we have to force it to make a change. So just doing something like changing the display adjustment slightly will force it to rebuild that render. Okay, so the display adjustment palette can be useful just to get the shadow projection to just rebuild for you. Okay, so you can imagine this is all demo data we're working with. These are tiny, tiny data sets, and you can see a progress bar. So imagine a huge data set with this would take considerably longer. Okay, so shadow projection, I currently have my background set to black, which doesn't really show it very well. So you don't have to follow along with me right now. I'm going to change mine back to gray just so you can see that a little bit better. Okay, so I just changed my background to gray. That will help you to see shadows if there is any based on your light source direction. Okay. Okay, so I just wanted to show you where all those controls were, how you can adjust how it looks and optimize it for whatever your data set is. Okay, any questions so far? Are we good? Good, okay. Next thing we're going to look at is we're going to take a quick look at surface segmentation. Okay, now we're going to do this on 2 different data sets. We're going to do it on this one, and then we're going to switch to a different data set. But we're going to introduce objects to the 3D view. So to do that, you're going to see a set of very small icons that are listed above your Surpass list. Okay, so these little icons at the top of your Surpass list. Some of these icons may look familiar to some of the icons I had in my talk this morning. These are the icons that represent the models that we have in Imaris, as well as a couple of other ones too. So the first one we're going to look at is the surface model, which looks like a little blue blob. Great description. Blue blob. Now, if you put your cursor on that little object, it will come up and tell you add new surface. So you can confirm that you're getting a surface object. So I'm going to click on it, and it will add that into the Surpass list. Okay? Once you've added that in, I do want to just tell you that these objects can be renamed because Surface 1 is not meaningful. And as you get to start using Imaris more, you're going to create multiple objects. So you can name them based on the cell type that you're identifying or the structure that you're trying to identify. Okay? So if I double click on this, I'm going to call this one Vessel. Okay, so now I'm using a surface to represent a vessel, and I'll be able to, when I save this and open it up, I'll know exactly what I was doing with this. So down at the bottom left-hand corner, we have a configuration. We call it the Creation Wizard. So the Creation Wizard is a set of steps that guide you through how to create one of these objects for the structure you're trying to find or detect. So the very first window or the very first step asks if you want to segment a region of interest. Regions of interest are important if you only want to measure a particular part of your data set, or if you have a massive data set and you want to only look at a portion of it. Okay? The other nice thing with the region of interest is it can be used temporarily, meaning that you can use it to set up your parameters, and then once you've set everything up, it will apply it to the entire data set. So the little option that says segment only a region of interest, I'd like you to click on that. And you'll see another option right underneath it called Process Entire Image Finally will pop up. And that's the option that if you have that selected, it means that it will temporarily use a region of interest just so you can set up your parameters, and then the entire data set will be processed at the end.
Can you select that after? Like if you change your mind that you want to actually...
Absolutely, yes. So the question was, can you change that? So if you change your mind, you don't want to use a region, can you go back? So down in the bottom corner here, there are 4 buttons. One is a back arrow, one is a forward arrow, one is a green finish arrow, and one is an orange cancel. Not an arrow, button, sorry, not arrow. And these allow you to navigate through the Creation Wizard. So if you go forward and then you're like, I don't want a region, and you want to go back, you click on the backward arrow.
I mean, so if you do the region and then you don't click to use it for the entire image, can you do that after?
Yes.
Okay.
Yes and
so
yes.
this button's still there the whole time?
Yes.
Okay, cool.
Yes. So in this case, we're going to use it, but we're going to process the image finally. So that I would like you to select both of these. And we're going to go to the next step. Now we're going to click on that blue arrow at the bottom, and that takes us now to step 2. Step 2 is setting up your region of interest. Now, you should have something that looks a little like my image here, which is basically a box around your image with little arrowheads on each face of that cube. Okay? Those arrows allow you to change the size of the region of interest. So, notice that you can't do anything when you're in navigate mode. So this is where you want to tap your escape key. If you're in the select mode, you can grab those arrows and adjust that region of interest. Okay? So this is where keeping your finger on the escape key is useful because I'm rotating and I'm then switching in and out of 2 different modes. Okay? This is really important with something in 3D because you are adjusting your region not only in X, Y, you may also be adjusting it in Z. Okay? So I've chosen, I'm going to choose a region over here, like so. It doesn't really matter where you put the region. This is just an example. Also, there is an option if you wanted to do multiple regions. So maybe you have a big data set, but you really only want to focus in on a couple of regions. So there's a little add button here, which will allow you to add a new region of interest. There's also delete if you choose not to. Okay, so I'm going to go to the next step, and this is where I get to choose my channel and my thresholding technique. Okay, so I'm going to choose channel 1, or the red channel, and the second thing I'm going to go through with you is some thresholding. So Imaris provides 2 different thresholding techniques for a surface. For those of you that have had a one-on-one session, or those of you that have used this before, the reason we have 2 is to optimize your segmentation. The first option, which is the default, is called absolute intensity. Now, absolute intensity allows you to choose one value that identifies the cutoff between background and signal. Okay, one value. So that means if you have one object, or all of your structures are the same intensity, absolute intensity is likely to work well. It's not common though in biology. You have typically cells that are different intensities, or structures that are different intensities. So if you use absolute intensity, it's going to struggle to find one value that works for everything. Okay? So the second option is called background subtraction, or local contrast. What this does is it asks you to give it an idea of how big the structures of interest are. It applies a background subtraction temporarily to the data and applies a threshold to that background subtracted data. Okay? That means that it's basically increasing that difference between background and signal, so it's easier to find the boundaries of where your structures are. Okay? So I'm going to show you an example of absolute for this structure, because it's one structure, and then the next data set will do this with the background subtraction option. Now, one last thing. This surface detail option, most of the time the default value will work fine. Sometimes it will cause a problem, so I need to explain it. Basically, that value in that box is equivalent to 2 pixels in diameter. So we apply a 2 pixel smooth to the surface itself. The reason for that is your data is made up of a series of cubes. So the very edge of your structures is a jagged edge of these boxes, effectively. Okay? If you apply no smoothing, the surface will be very rough. It takes more triangles to represent that. It takes more time, and it also doesn't look very good. So a smoothing filter is applied by default to make the edges look smoother. If you are working with tiny puncta, tiny little structures, that smoothing filter may prevent you from actually putting a surface on those small structures. So if that happens, turn it off and see if you can see a surface. Okay? Also, for those people who have maybe looking at nuclei and there's variations of intensity within a nuclei, you can apply more smoothing that kind of blends out that surface boundary a little bit better. So it can be used in multiple different situations, but the default value for probably 80% of what you're doing is going to be perfectly fine. Okay. So for right now, we're going to go to the next step, and this is where we have a little threshold. Okay. So we have your intensity profile. So you have intensity running along the x-axis, and then we have frequency on the y-axis. This yellow range here is everything that is within threshold. Anything that is out with that is out of threshold. So this stuff would be your background, and this stuff is the stuff of interest. If you grab that little orange slider right there, you'll see that that threshold will change interactively for you. Okay? So depending on what it is you're trying to segment, you can adjust this as necessary. Okay. I'm going to come back to this split touching objects option for the next data set we look at. But for right now, I just wanted you to see that that is an interactive preview for the surface. I'm now going to go to the next option, and this is where you can classify. So if you have any little blobs, little structures, cells that you're not interested in, something that you want to get rid of, you can classify surfaces based on any of the statistics we calculate. So that could be size, or intensity, or position information. Okay? Again, we'll come back to this one in a moment when we try it with a different data set. But for right now, I'm going to go ahead and click on the next button. And you'll see that it says complete ROI. So basically, I've finished all of my parameters, and now it's ready to apply this to the entire data set. So I'm going to go ahead and hit the green finish icon, and it will apply it now to the whole retina. to the whole vessel. Okay? If I turn the volume object off, that will let you see just the surface model on its own. Okay? So you'll be able to see something that looks very similar to what your blend mode looked like. Okay? Now, one other thing I want to show you. The light source that you were playing with earlier also works with a surface or with any of the models we create. So if you notice that your surface looks a little dark, it might be that your light is oriented in a weird position. So go click on your light source, make sure you're in select mode, and you'll see that in changing that orientation of light will impact how your surface looks. Okay? Okay, so last thing before we switch to a different data set. Once we have created a surface object, you will see a list of tabs along the configuration window, and these tabs allow you to do certain things. One of them looks like a little graph or a plot. That is your statistics tab, so that's where all of your measurements are stored. So whether you want to or not, when you create a model, it automatically calculates statistics for you. Okay? So there is an overall tab which lists the number of objects, and there's also a detail tab which gives you the object by object statistics. Now in this case, it tells me I have 4 objects. Okay, so you'll see I have the vessel, and then I have this blob, and this blob, and this blob. Okay, so if I go to the detailed view, I also have 4 separate measurements. And right now, for everyone, you should see that it's giving you the area. Area is our surface area. All of the statistics are listed alphabetically. So if you click on where it says area, you'll see all of the different statistics that we calculate. Okay? So if you go all the way to the bottom and click on volume, now you can see the size of each of those structures as a volume. Each of these, if you click on them, will highlight the structure that the measurement pertains to. And the same over here as well. So remember, select and navigate. If you're in navigate mode, you will not be able to click on anything. If you're in select mode, you'll be able to select objects of interest and click on them. And you'll notice it will show you the measurements over on the left-hand side. Okay? So I'm going to show you one other thing. If we select multiple objects, I'm going to hold the control key down. That will be the command key on a Mac, I think. You can try it. Will it let you select multiple? Is it command or control? Command. Okay. So you can select multiple objects. You can also, I'm going to show you one other tool. There is a little tab that looks like a pencil. That is your edit tab. And if you click on it, oops, wrong one. If you click on that edit tab, there's an option in there to delete. So if you go in and make a selection on multiple objects, and you want to get rid of them, you can delete them in the Edit tab. Okay? So, one last thing. The statistics, incidentally, can be exported out of Imaris using the, so when you're in the Statistics tab, in the bottom corner of that configuration window are 3 little icons. 2 of them are probably, sorry, one of them is grayed out, but they look like little save disks. Okay? The single one will save the statistics you're currently looking at, that volume table, and the multi-disk icon will save everything, all of the statistics we calculate. So I'm going to go ahead and click on the All Statistics, and I'm going to save it. You'll see that you've got comma-separated values, and you've also got Excel, either an XLS or an XML. That XML, for those of you who are using Imaris right now and generating a ton of statistics, that XML is there as a workaround for a limitation of about 65,000 rows in Excel. So you have a maximum number of rows of measurements in Excel of 65,000. If you exceed that, it won't save the file, so XML is another option for you. Okay? I know that sounds like a lot, but you hit it so fast with that. Okay, so I'm just going to go ahead and hit save because I want you to see what this Excel spreadsheet looks like. We separate out your statistics into separate sheets. So this is very simple because I have one object, but you'll see down at the bottom on my screen, I don't know if you have Excel on your in your Mac, so I don't know if you can do this part, but basically every single statistic is listed as a separate sheet, and all of the objects will be listed on the in each of those sheets for each object. Okay. Okay, so we're going to change it up a little bit. So we're going to go back to the arena view. And it's going to come up and ask you if you want to save any of the changes. I'm going to say no at this time. And I'm going to go and find another data set called plant cell. So it's this one right here. It looks like a cell with little green chloroplasts inside of it. Has everyone got it? Yeah, okay. So if you go ahead and open that one up, this one's got a little bit more going on. And I'm going to show you a little bit of object splitting as well as a different thresholding technique for surface, which I would think the majority of you will probably end up using. So I'm going to turn off the cell wall for right now, and I'm going to focus on the chloroplasts in this plant cell. Okay? So we're going to go in and make another surface object by clicking on that blue blob. I'm going to give it a name. I'm going to be lazy and I'm going to call it chloro. And I'm going to go through the creation wizard for this. Okay? I'm not going to do a region of interest this time, so it's going to take me straight to find the source channel. If anyone needs any help, please let me know. Just wave your hand, let me know. Give me a strange look. Somebody just waved their hand outside the door, so that was awesome. I'm going to go to channel number 2, which is the green channel. I'm going to leave the surface detail as it is. Remember that default value is 2 pixels in size. And I'm going to change the thresholding technique to background subtraction. Now, the value in there is a default value. I'm actually forgetting right now how it's calculated. It's a certain number of pixels, but it's a default value, so it's not one that I would recommend you just use. It probably won't work. So here's how we're going to figure this number out. So it says here, diameter of largest sphere which fits into the object. An easier way of saying that would be, what's the size of your smallest structure? Okay? So if we go to, up at the top here, you'll see we have a window or a button called Slice. It's right next to the 3D view window, or icon. If we click on this button, it will take us into our slice view, which lets you look at individual slices for that data set. And you can scroll up and down using this slider on the left-hand side. Okay? If you find a particular plane where you can see the chloroplasts, you'll notice that if you click on the image, it will add a little cursor, a little mark. And if you click again, it will actually draw a line in between those 2 points. So this is a quick and easy way to get an idea of the size of the structures you're working with, okay? You're going to use this a lot because in a lot of cases, people don't always know how big their cells are. Or at least they think they do, and then they measure them and it's not actually the cell size. So this will give you an idea of how big it is. So the value or the size of that line, the length of that line, is over on the right-hand side, listed as a distance. So this chloroplast from edge to edge is about 5 microns in size, okay? Now, you want to try and find one of your smallest structures because we're doing a background subtraction, so I want to make sure that I put in roughly my smallest size. So in this case, I'm probably going to go with around about 4 microns. So I'm going to type in 4 microns right here, and I'm going to go to the next step. Okay, hopefully everyone sees something that looks similar to what I'm seeing right now. Okay.
How do you exit from the slice view?
So you just click on the 3D view.
Oh, 3D.
Yes. So it's basically just a different view. So if you click on slice, just click back on 3D. 3D view is that that work area for your 3D rendering.
Thanks.
Okay. So if everyone's done this, I'm actually going to ask you to do something a little different. I want you to go back a step. And I want you to turn absolute intensity on, and I want you to go forward. And I want you to tell me if you see a difference. Okay? So hopefully what you guys see is that if you use absolute intensity, a lot more of those chloroplasts seem to fuse together. Okay? That is the big issue or the big problem with people using absolute intensity, because you're using one value. So remember, if you have a really bright structure, it has a residual amount of fluorescence around it, which is dimmer. And that could be very similar to the intensity of some of those dimmer structures in your data set. So as you bring down the intensity to get the dimmer structures, you start over segmenting the brighter structures. Okay? Background subtraction normalizes that for you. So I highly recommend, for the majority of people that I work with, background subtraction is the preferred method for this. Okay. Second thing we're going to do in here, and also you still can adjust if necessary. You can adjust the threshold, but I'm also going to look at the split touching objects option, because this allows you to apply a mechanism to find your objects and then use a watershed filter to split them apart, because you'll see a lot of these chloroplasts are very, very close together. And you'll see that as I try and fill out to the edge of those structures, they kind of fuse with neighboring structures. So I'm going to enter in what's called a seed point diameter. It's exactly the same parameter that I put in when I was telling it my background subtraction value. So it's really looking for the smallest structure size. Okay? Because what we're going to do is we're going to place a spot or a sphere inside each of your structures. It's then going to use that to apply a watershed filter to basically figure out where we're going to break them or split them. So I put in a value of 4 down in the seed point diameter, and I'm going to go to the next step. And hopefully what you're going to see is a spot or a sphere that's 4 microns in size in the center of each of those chloroplasts. Okay? You do have a quality filter in there, so if there are some chloroplasts that are not getting picked up, you can reduce that quality filter to find those dimmer intensity structures. Okay? Now, there may also be a case where, and I'm going to show it right here, do you see these 3 chloroplasts? 2 have spheres in them and 1 does not. Okay? Even if I bring this tolerance or this quality filter down, it doesn't matter. I cannot get a sphere to go in that 3rd structure. So the problem here is that this sphere is overlapping with basically the bound or the edge of the other object. So what we're looking for is, when you have a peak intensity, we're actually looking for a drop, so we can tell where that split's going to occur. If the sphere is too big and covers that drop in intensity, it won't be able to place another sphere next to it. So if you have this happen, go back a step and reduce the size of your sphere. It doesn't have to be by a lot. I just made it 3.5 microns. And you'll notice that in doing that, it now placed another sphere next to it. So if things are very close together, and your sphere is too big, it will have a hard time splitting. So you need to get that sphere size correct. Okay. Now, one other thing I'm going to tell you, there could be an instance where you have, I can't see any, but maybe you guys do. You may have a situation where you see 2 spheres in one object and they're overlapping. Okay? Don't worry. That happens. You can have multiple peaks inside one cell. If they are overlapping, it will basically see them as one structure. Okay, so it won't split it. So if they're overlapping, it won't split. Okay, so I'm going to go ahead and I'm going to go to the next step. And this is where the surface goes through this watershed filter, and hopefully you can see these little cracks in between the structures. Okay, now instead of just getting one big connected object, we're going to have separate objects, so you'll be able to get more information on size and intensity for each of the structures, okay, and counts also. So we get to the point where I'll zoom out a little bit, where we have a lot of structures now, and this last step is called classify classify surfaces, okay? This is where you get to choose what criteria makes up the object that you're interested in, okay? Maybe you want to specify that your object has to be a certain size. So you'll see that as I'm adjusting this filter down at the bottom, it's hiding and showing structures that don't fit into my classification. So the default one is called number of voxels. It's effectively an uncalibrated volume. Okay? So you'll see on that little histogram, it's basically showing you frequency and versus size or number of voxels. So the stuff up over here on the right hand side is the big structures. The stuff on the left hand side is your small structures. So if you were to drag that cursor or that threshold on the right hand side, that gets rid of the bigger. And if you drag it on the left hand side, that gets rid of the smaller. Okay? One other thing. You have to right click to activate the upper threshold. Left click to get the bottom threshold. Does that make sense? Because if you try and left click on the top threshold and it's not activated, it will bring your minimum threshold all the way up. Okay, so right click to activate the top threshold and left click for the bottom. Okay, I'm going to show you a couple of other filters that are useful. There's one in here called distance from image border, which allows you to get rid of anything that touches the edge. So if you're counting and you don't want to count half a cell or half a structure, you can actually adjust a boundary or set a size or distance in microns from the edge of the image that will be excluded from objects. Okay? So you can actually, typically people put in a cell diameter. So if you know your cells are 10 microns, you'd probably use a 10 micron distance so that you exclude anything that could be a portion. So in this case, I'll probably type in 10, and you'll see that in doing that, it gets rid of some of those chloroplasts that are close to the edge. Okay. Okay, also, lastly, this is an additive filter. So you can actually have multiple criteria that make up this object. It could be a size-based filter, and then a distance to edge filter, and then an intensity filter. So these are additive filters in here. Okay. Lastly, I'm going to just basically include everything at this point. I'm going to hit the finish button, which will give me my surface. I'm going to turn my volume off just so we can see that a little bit better. So here are the objects that have been split, or the chloroplasts that have been split. I'm going to show you quickly the statistics tab that we looked at earlier now has a big list of measurements because there are several objects. In fact, if I go to the overall tab, I have 239. Okay, so there's your count. In the detailed, you can get a lot of information: volume, surface area, longest principal axis, intensity values, whatever you want. But I'm going to show you something else. There's a little color wheel on that surface kind of list of tabs, right next to the statistics tab. This is where you can apply statistics color coding. Okay, so by default, we color the surface based on the channel that was used to create it. So if it was a green GFP channel, it will be green surface. Statistics color coding allows you to color code based on any of your statistics. So in this case, I'm actually going to color code based on size. So I'll go down to volume. So the cooler stuff is smaller, the warmer stuff is bigger. You can also do it though based on distance. So if I go for the distance from border, you'll notice everything in the middle is warmer, meaning it's a further distance from the edge, and things next to the edge are cooler. So statistics color coding can be really useful for visualization. Okay, how are we for time? We're not great. So we have about half an hour left. So I wanted to just get this concept over because we're now going to start doing some other things that are going to use Creation Wizard. So I'm hoping that this is making sense so far because I'm going to start speeding up to crush it all in right now. Okay, so I want to show you animations real quick using models or surfaces in this case so that you know how to create movies. So we've made a surface, we have statistics, I put some statistics color coding on there, and what I'm going to do now is I'm going to turn the surface off and focus on the maximum intensity projection. I'm going to go in and switch into blend mode. Doesn't look too good right here, so I'm going to make some adjustments to my display and also to my blend. Okay, so if you guys can follow along, please do so. So I'm just making some changes to how that's visualized. I'm also going to bring up the brightness a little bit for the chloroplast. So here's now a blend mode for my plant cell. Okay. there is a button down in the bottom right-hand corner called Reset, and Reset will take you back to the original XY orientation of your image data. So if you've kind of rotated and zoomed and you just want to get back to that known position, the bottom right-hand corner, hit that Reset button down here, and that will take you back to your known position, okay? Now, the reason I'm setting this all up to look a particular way is we're gonna go in to make an animation, and I wanna start off like this, okay? You don't have to, but in this case, I'm gonna start off looking like this. So I'm gonna click now on the top bar. So up near your 3D view, you'll see there's an option called Animation. If I click on Animation, it will add in a keyframe animation bar at the bottom of your 3D view, okay? So down at the bottom. In this animation, or keyframe animation bar, there is a rectangle with little blue vertical lines in it. That is actually, or those are what we call animation frames. Those little vertical lines are representing animation frames. And what we're gonna do is tell it how we want this animation to go, okay? So the first thing I'm gonna do is I'm gonna click on this Add button, which is over on the left-hand side of that keyframe animation. So click on Add, and you'll see it will put a thick white bar in at the beginning and also a thick bar at the end. The reason we actually do one at the beginning and one at the end, sorry, am I blocking you? I'm so sorry. We do one at the beginning and one at the end because we want the first frame and the last frame to be the same for the purpose of looping, okay? We can change that, but that's just how we do it by default. Okay, next thing. I would like to rotate the plant cell like so. So I'm just gonna turn it from one side to the next. Doesn't have to be perfect, but now I'm going to click on the Add button, okay? Now when you do that, you'll notice it puts another frame right in the middle of those animation frames, okay? And now I'm going to turn it to look through from the other axis and click on Add. And again, it's chopped up your animation frames and put in another frame, okay? So what I want you to do is go back to this very first thick bar. So these thicker bars are what we call keyframes, okay? They store location, rotation, and also object information, okay? So if you click on it, you'll notice your image will go back to the original position we were in. And if you hit the play button, which is right over here, if you click on play, you'll see that your structure will move between those different keyframes. So every frame in between two keyframes becomes a transition frame. So they basically interpolate from one position to the next, and then when you get to that new position, it then interpolates to the next, okay? So instead of just jumping around, you're transitioning between those. Okay, so we're gonna make it a little bit more interesting. So I'm gonna go to that third one that I just added in and select it. So my third keyframe. I'm now going to add in my surface, and I'm going to turn my volume off, okay? And then I'm going to click Add, okay? Now I'm going to zoom inside of my plant cell, like so, and then I'm going to click Add. And then I'm going to just do some weird stuff like rotate around and look from the other side, and click Add. So every time I'm making a change, I'm clicking Add so it remembers what I'm doing, okay? I'm also going to show you another cool tool with surfaces, which is called transparency. You can make your surfaces transparent. So if you go to your surface object, you'll see if you go into the color tab, there's a button at the bottom called transparency, and you can actually make your surfaces transparent. That can also be included in an animation. So now I'm going to click Add, and I don't know, I'm just gonna rotate a little bit more, and then I'll bring the transparency back down and click Add, okay? So now if I go back to the start and hit the play button, you'll see that it will, actually you don't have to start from the start, you could start from wherever you want in the movie, but you'll see that now it's basically providing this progression through whatever it is you want to present, okay? Now, the default number of animation frames is usually 200, or maybe it's 100, I forget now. It's either 100 or 200. 200, okay. Depending on what you're doing, if it's a very simple rotation, 200 may be too many frames. But as you add in more keyframes, you're actually making the animation more complex. And so you're probably going to need to have more transition frames, which means you want to increase the number of animation frames. So that instead of it, you know, transitioning between two keyframes with only a few animation frames, you're basically adding in more transitions, okay? Smoothing out that transition. So all of this is now, we're kind of configuring it for exactly how we want it to look. I'm going to add in one other thing, and I talked about it a little bit this morning. So I'm going to go to probably the second to last keyframe, and I'm going to add in another object to my surpass list, and it's the one that looks like a pair of scissors. Hopefully everyone sees that. So it's a little bit across from surface. It looks like a pair of scissors. If you add that in, you'll notice it's called a clipping plane. Okay? Clipping plane is this tool that allows me to hide and show different parts of the image. And it has this manipulator tool that allows you to decide how it transitions and what orientation it clips. Okay? So let me show you this real quick. Notice in navigate mode, you won't be able to do anything but move the data set. So be in select mode with the escape key. If you grab the thicker part of the manipulator, you'll see that that will allow you to adjust its depth. Okay? If you grab the thinner part of it, you can change the orientation in which you are slicing or clipping a particular plane. Okay? So the two of them together allow you to decipher the orientation and the depth, or the depth of the plane. Okay? Also, there is some controls for clipping planes. So if it's clipping and you would like it to clip the opposite direction, there is a flip option. So flip will basically change the direction in which it is clipping. You can also change it to a preset orientation, X, Y, X, Z, Y, Z. Okay. Last thing, there is an option to show the manipulator. There's also an option to not show the manipulator. Most people would be kind of weirded out if they saw a big white manipulator in the middle of your Imaris movie in a presentation. So I would probably recommend you turn it off. But let me show you how we can incorporate it into a movie. So I'm going to start by putting it in, but I'm going to back it off so it's actually not clipping anything. And it doesn't matter what orientation you're in, it's no big deal. I'm going to turn the manipulator off. I can do that in the configuration for the clipping plane. I am going, oh, and then also you can turn the frame off. That's that yellow part that shows you it's being clipped. That's up to you. I'm going to go ahead and hit the modify button because I'm actually going to modify the keyframe I'm looking at right now. Okay, so just to make sure I'm looking at the right one. Okay, so I'm looking at this keyframe. I'm going to click on the modify button, and now I'm going to turn my manipulator back on, and I'm going to drag it through my data set, turn my manipulator off, and then hit add. Okay? Then I'm going to turn my manipulator back on and do this, and turn it off, and click add. Okay? So if I go back to this frame right here, or maybe it's this one here. Oh. Am I clipping it from the, oh, yes. Okay, because I added it later, you'll notice that it's actually clipped everything. So make sure you go back to your first frame and make sure it's turned off. So I'm going to look and see where I clipped it back in. I think I was good here. Aha, okay, so I must have started it around here. Okay. So I'm going to go to the last frame where I'm not clipped and hit play. Oh, that kind of sucked. Okay. Ah, okay. So I did something a little wrong. I don't think I modified, so I'll go back and change it. But here you can see you can combine rotation and clipping and all of this good stuff. So clipping planes also, there's an important part of this. If you add a clipping plane while you're making a movie, it will leave it on for other frames. So make sure you go into your first frame, turn it off, and click the modify button so it always remembers that it's off for those other frames.
Quick question.
Yes.
So I have a scale bar and a color bar, but there's no labeling. I'm curious, it's 20 microns.
Mm-hmm.
How do I get a label on mine?
Okay. So we're talking about this little label. So this is a scale bar down at the bottom. I'm going to move it so people can see it. You need to be in select mode to move it. So this scale bar is on by default. It can be modified for position and label. So let me just show you real quick. You can change the size of it. You can also drag it from the bottom and make it thicker or thinner. If it does not have a number on it, go into your preferences under the file menu, go to display, and there is a whole section in there that allows you to configure what your scale bars look like. So there's one in there that's called show scale bar label. It should be turned on.
Okay, yeah, mine is on, but I'm not seeing any numbers. That's okay.
I'll have to look at that. Is that the same for everyone incidentally, or is that, I'll have to take a look at that later. But it should be there, and it should be configurable in this little window here. Okay, so okay, just back to the movie. I want to show you how we save a movie. Oh, one last thing about clipping. Go for it.
So, like, does it measure, like, from your metadata, can it take, like, the actual size, or do you write the 20 micros bigger?
So remember we were talking about metadata previously? So basically, we read your pixel resolution from the metadata. So the calibration bar that's in there is usually, if your metadata is there, it'll be calibrated for you.
Okay.
Now, as far as size is concerned, if it's a brand new installation, usually it's a 10 micron scale bar. If you want to have it so that it always defaults to the same size in the preferences, there under display, there's an option that actually allows you to decide the size of the scale bar.
So when you said you're going to make it larger or smaller, then, like, you still have 20 microns there, but... Like,
No, it doesn't. It changes the value. So as I make it bigger,
Oh, okay, I see.
yes,
I didn't see
if I make it bigger,
see.
it will
Okay,
change
it's
the
one of those
size.
three.
Yeah. And you can also make the font size bigger by clicking and dragging, and you can make it bigger so it's a little bit easier to see. But no, that scale bar will change. It will also change as you zoom. So if I'm in navigate mode, you'll see that as I zoom in and out, because you're now looking at a structure at a different zoom level, that will also change with you. I do have some advice for you though. If you're incorporating zooming in a movie, that gets really distracting for people because they see this like number changing and they're like, what? So typically I would recommend for movies, either just have a scale bar with no label on it, but also be prepared that it's going to change in size because that 10 microns is going to get bigger and smaller. My advice is actually to turn it off altogether for animations in particular. So, but if you have to have it anything, have it with just the bar, not the number. Okay. Okay, so the clipping plane, there is an important piece of information with the clipping plane. Clipping plane will clip everything underneath it in the list. So the position of the clipping plane makes a big difference. So if, for example, you don't want to clip the frame, so you clip all of the data, but the frame stays there, make sure the clipping plane appears underneath your frame, or move the object you don't want to clip above the clipping plane. Okay? So right now, I have my chloroplast surface turned on, but I can't see it because my clipping plane has actually clipped it out. So it's actually here, it's just being clipped out. But you'll notice that this is kind of cool because you can actually have other data stay visible, but remove others. So this could be kind of cool if you're trying in a movie to describe that you have multiple cell populations. So you can use a clipping plane to remove one of them to leave the ones that you want to show. That's one example. Okay. Now, as far as saving a movie is concerned, once you have figured out how you want your movie to progress, also notice there are delete keys. So if there's a keyframe that just isn't working for you, you can delete it. You can also space the keyframes apart at different levels. So if you want something to take longer to transition or shorter to transition, you can move the keyframes around as well. So once you're done, hit the record button on here. And once you hit record, this will open up the save dialog. The default format for saving is the H.264 codec for MP4. It's kind of most universally, like, we had so many issues with people with Macs and PCs and PowerPoint and trying. This format will work with everything as long as you're using current. So if you're using like Office 97, maybe not. But if it's a current operating system and a current version of PowerPoint or whatever you use, this should work fine. Okay. Just so you know, though, we also provide a raw movie, an AVI that is uncompressed. That's massive. So if you're using that format, be prepared for it to be big. We also have a TIFF series. So a lot of people use VLC or other Handbrake. I saw Adam using the other day. If you have another movie maker that you prefer, or if you have another codec you want to use that we don't provide, export your frames out as TIFF files, bring them into your movie maker, into iMovie, whatever it is, and you can use whatever codec you want. Okay? In this case, I'm going to use the MP4. It asks you for frames per second. I'm probably going to speed mine up a little bit and use 10 frames per second. Also, the default size of the movie. Oh, sorry, yeah.
Oh, so just a quick question. So we have this experience of like when we make a movie, sometimes it just make say in Windows and then it doesn't play in Mac, and then in like say if we make in Mac, maybe it doesn't play in Windows. So do you have a suggestion of like how to work, like what's the workaround of this, like which format is like basically useful for both?
Okay, so typically AVI is the one I would recommend for both. However, for QuickTime, you actually have to install a whole nother set of codec for AVI for Mac, which is a pain. So the one that works the best is an MP4, but I'm going to be honest with you. The codec we had in version 8 and 7 for MP4 used a lot more compression, so the resolution was not as good. Imaris 9's codec is a lot better. But for you doing it right now, my suggestion would be AVI, and make sure you have the right plugins for QuickTime. Okay. Okay, current window size is the default size. If you want it to be a preset size, 1024 by 1024, or a particular resolution of your screen, you can choose from one of the presets. I'm just going to use window size right here, and I'm going to go ahead and hit save. And what it will do is it will actually play through the movie as it's creating it, so you can see what it looks like. Okay, I still have quite a bit to do, so we're going to jump really quickly into some co-localization. Okay, I'm going to use it with this data set to demonstrate one part, and then we're going to switch to a slightly different data set to do another part. So, okay. Oh, my manipulator's in there. No! Okay. It's okay. I'll survive. Okay. So, let's close this down. Now that we're done with this, I'm going to turn the surface off, and I'm going to take you, so let's put the volume on, and I'm going to go back to maximum intensity projection. So I'm kind of resetting here a little bit. At the top of your screen, you will see a little dialog called Colloc. In Imaris, we provide two different mechanisms to do co-localization. One is an intensity-based approach, which is available on Fiji and lots of other applications that provides co-localization statistics like Pearson's correlation, Mander's coefficients, percentage co-localization, etc. We also have another approach that we call an object-based approach, which is where you identify the objects of interest and look to measure their interaction or their distance to one another. I would say more nowadays we're seeing people use an object-based approach, but I'm going to start with intensity and then we'll switch to object, okay? So the intensity-based approach is this Colloc button at the top of your screen. So what we have in here is a slice view of your data. We have a threshold for channel A and channel B. You can choose the channels. If you have more than two, you can go in and choose which channels you want to use. And these are thresholds. So basically for colocalization, you need to set a threshold. Anything above that will be considered in your analysis. Anything below that will be completely ignored. And we also have a little scatter plot here just kind of showing you frequency. So ideally in a world of colocalization, you would want a little 45-degree line, but no one ever sees that, so perfect world. What I want to show you here is ideally what you want to do is select your thresholds for channel A and channel B. You can do that manually. You can type in a value over here, but you can also use a tool in this window that allows you to draw a region around the structures of interest. And what it does is it extracts the minimum intensity value within that region and sets that for your threshold. So on a Mac, I'm not 100% sure if the keys will be right here, so bear with me. If you hold down the Control key on your keyboard and click on channel A, which in my case is red, it should put a little region of interest around the area that you are clicking on. Is that working? It's Command. Sorry. I keep forgetting that. Now, it should be for channel A, so if you hit the Command key and then click and drag, that will make a little lasso, basically, a region around an area. And as you may have noticed, it changed your threshold for channel A, okay? I'm now going to go in and do the same thing now for the green channel. I'm going to use the Shift key for the channel B. So I'm going to go in and find a dimmer object that represents B, channel 2, or the green channel. And I'm doing the same thing. So now I've got a little region around red. You ideally are looking for the lowest or the dimmest signal that you are still interested in to set both A and B. Once you do that, you're going to start to see little red overlap regions appear in your data. Is anyone seeing that? Little white regions. Those white regions indicate regions that are positive for colocalization. So basically means that those regions have green and red intensities above thresholds in the same voxels. Okay? Now, over on the right-hand side, we have a little estimate of your colocalization statistics, but there's one other thing I need to show you. Typically, when you're doing colocalization, you want to make sure you're only looking at colocalization either in a particular cell, or in a vesicle, or in a structure of interest. So what we're going to do is we're going to activate this option at the bottom called Mask Dataset. Okay? If you click on it, you can choose a channel that you're going to use as a mask or as something that's going to define the structures of interest for you. In this case, I'm probably going to do the chloroplast, so I'm going to switch to the green channel. And what you're going to notice is if you click on that threshold, you're going to start to see that as you change that threshold, you're going to see this hatched background appear. It's hard to see on my screen, but hopefully you guys can see it. So I clicked on this little threshold here, and as you move it around, you'll see that as you drag it up, it's going to basically mask out more, and as you drag it down, it will mask out less. Now, because I'm using the green channel, you should be looking at the chloroplast to define where that boundary is. But anything that's in that hatched region is going to be ignored from the colocalization analysis. So your background is being completely removed. Okay? So once you have done that, over here you have a breakdown of your colocalization statistics. All of these are detailed in our reference manual. There is also a colocalization tutorial on our website, so you can go in and see how this is all done. But one thing I wanted to show you is that it will give you a breakdown of the percentage of colocalization within your region. Now, your region of interest is the unhatched area. Okay, so right now I only have like 4% or something very small, which doesn't surprise me. This isn't a true colocalization data set. And then it also gives you the amount of colocalization for each channel. So it breaks it down to a percentage. So of all the red intensity above background, how much of it is green? And for all the green, how much of it is red? And that's what those two other percentages are. Okay, one last thing, and then I'll show you the object-based approach. Over on the right-hand side, there is a build colloc channel. So once you've set everything up, it will generate a brand new channel for you. So if you look in your display adjustment, you'll see a third channel called colocalization result. If you go into the 3D view, you'll be able to turn off the other channels and see where the colocalization occurs, okay? And also, if you click on the colocalization result, like the name of the channel on display adjustment, it will open up your colocalization statistics. These can be exported out as a CSV from this point, but it will also get saved with your data as well. Yes?
I have a question if you can do colocalization within a region, like say you use the cell function and you're interested in cells that have, say for single molecule fish or something, transcript A and transcript B in one region, and then
Yeah.
mask those out
Yeah.
into ones that don't
So you
have
can
it.
do it in a couple of ways. You can use the masking feature here. One other thing you can do is actually use a surface to basically put a boundary on the structure of interest, like your nuclei, and mask the outside to black, make it 0. And then it's effectively the same thing. So you can do it in multiple different ways. That way it's just kind of doing it all in one dialog for you. But yeah. Okay, next, I'm going to switch to a different data set. So I'm going to click on Arena. I'm going to say no. And I'm going to find the object that the image that's called PTK2 cell. I had to say that slow because I mess it up all the time. Okay, so it's a 4-channel image, yeah. So we basically have 4 different channels representing DAPI nuclei. We have, chromosomes. We have, kinetochores, microchip. We have a bunch of stuff. What we're actually going to do is we're going to turn off the DNA channel with the blue. We're going to turn off the microtubules, and we're going to focus on the white and the red blobs. Okay? Now I'm going to show you the spot detection tool, which we haven't really looked at yet, so I'm going to use this as an opportunity to show you how this works. And the first thing I'm going to do here is I'm going to use the slice view to measure the diameter of these white and red structures. So if I remember rightly, yeah, this is measuring about 0.27, so about 0.3 of a micron. I'm also going to measure one of the red structures, so measure here, about the same. Okay, so 0.34 in this case. So what I'm going to do is I'm going to go back into the 3D view and I'm going to create 2 spot objects, one to get the red stuff and one to get the white stuff. Okay, so if I go in, click on the little orange spheres icon, which is our spot detection. I'm going to call this red. And here's my creation parameter for this. I'm going to go to the next step. No need to do a region of interest here. And I'm going to specify my diameter as 0.3 microns. I'm going to go to the next step, and you'll see that in doing that, it's automatically put a little sphere in the center of some of those kinetochores. Okay? I'm going to use the quality filter to help me pick up some of the dimmer kinetochores in there. Okay? So once I've done that, I'm now going to hit the green finish button, and now I have my red kinetochores. For some reason, it should have changed color, but it did not, so I'm going to force them to be red. So I'm going to go into the little color wheel for my spot object and make it red. Okay. I'm now going to go in and do the same thing for the dynactin, which is white. Same thing. So I'm going to, oops. That was my bad. I hit the finish button before I was ready. So I'm going to go back in and do this again. So I'm going to go back through, choose the dynactin, change that to about 0.35 or 0.3, whichever. And to be honest, I think that's a little small. I'm going to use a slightly bigger value. I think I happen to... Yes, that's a little bit better. So now hopefully you can see I chose a value of 0.6. I think I must have measured like the top of them. That's a little bit more representative. I'm also going to bring the quality filter down because there's a few more that are a little dimmer that were not being picked up. And I'm going to go to hit finish. Now, one thing I'm also going to change, and this is also a good way to show you, you can modify your creation parameters. If you're not happy with how you have detected, you can click on this little magic wand tool in the one you want to correct and click on the algorithm option, and that will take you back in. So it will remember all the settings you used previously, but it will allow you to modify them. So I'm just going to modify and bring the size of these kinetochores up a little bit. So I'm going to use about 0.6. Okay, and then hit finish. So now I have two different spots, one for kinetochores and one for dynactin, and you'll see that some of them are connected and some of them are touching and some of them are not. So there is a tool in either one of your spot objects. It looks, a tab that looks like a little pink gear. It's called the Tools tab. So it's right at the very end next to your color tool. If you click on it, there should be a list of little functions or tools. These are called extensions. They're actually MATLAB extensions. So we have a two-way interface with MATLAB and Python. In this one, I'm going to choose the one that's called co-localized spots. And this one here is where we can actually ask it to find all of those that are within a certain distance to one another. Okay? It is assuming that because we only have two objects, those are the two objects we want to measure co-localization with. If you have more than two objects, you get to choose them. In this case, I'm going to confirm yes, red and white. Click OK. And then here's where I get to choose my distance. Now, depending on your application, you may have two structures that are very close, but very minimal overlap. So you would want to really use the center to center measurement. In this case, it would be 0.6. But you may actually want them to have some kind of overlap, so you may want to make that smaller. It's completely based on what you want. So in this case, I'm going to go with 0.6, so they at least need to be touching. Click OK. And what this does is it generates a new set of spheres. So I have 4 in here, 4 in a set called colloc. I have red collocated, red non-collocated, white collocated, and white non-collocated. And these are basically a breakdown of all of those spots, which ones are touching and which ones are not. So you can turn these ones off one at a time. So these ones here are representing all of the red or the kinetochores that are not, that are touching. And here are the white that they're connected to, and then vice versa. If I select the non-collocated, that will give me everything that's not touching. For some reason, I'm losing my color when I do that. I don't know if it's the same for you guys or not. It is losing the color. That's annoying. However, you can go back in and change that to the color it should be. That's really weird. That might be, oh no, they're there, okay. Not sure why it's losing the color. It shouldn't be. What I do want to show you though is if you click on the folder called colloc, go to the little statistics tab that looks like a plot, go to overall, and you'll see that it gives you counts for the number of structures that are co-localized versus not co-localized. Okay? Now, I'm almost out of time, and I didn't get to cover tracking. Okay, so here's what we're going to do. As I mentioned, I'm recording these sessions. So the next session, I will cover tracking, and I'll make sure that they get shared with you so you can get to see that. So I'm probably going to have to switch it around a little bit and do tracking before colloc for the next session so that we'll have one that does colloc at the end and one that does tracking. Okay. We're 5 minutes over, but does anyone have any questions before the next group comes in?
Does this also sort of work on, say, two surfaces?
Absolutely. So we have an extension called surface-to-surface colloc that can be used for that exact same purpose. Okay? Okay. Well, thank you so much. I hope that was helpful. It was a lot of information, but hopefully it helped you at least to feel a little bit more familiar with how to get started. And if you want to review this later, ask some of the people in the BIDC and they'll help you out with a link for this. Okay? Now I have a big favor to ask. Can everyone put themselves back in the arena view so that the next group know where to start? So you don't have to save anything that we've done. just go straight back into the arena view, and then we're all set for the next session. So thank you very much. Thank you. Okay. So was your system weird? Just missing, like, was it just the
Oh yeah. Yeah, it had the bar and it had the color.
Yeah.
Yeah.
I know.
Okay.
Yeah. And you're right, for movies it would look weird, but it just wasn't there. If it was simple toggles.
Yeah, it should be there. I have to tell you, I'm so used to using PC that whenever weird things happen,
Thanks for
I'm
telling
back,
me.
I'm like, so I'll report it. It might just be a little.
Okay.
Tell me how it was supposed to be again, because when I did it, it saved it as like a really grainy, it wasn't like the kind of, I don't know if it's.
So one thing to be aware of when you look at, I didn't save any of these as movies. Let me just do this. So when you go into animation, let me just add a couple of these frames and then go in here. There's this little icon, the slider bar compression. I didn't talk about it. If you touch that, it will actually, especially if you drag it over to the right, it lowers your resolution.
Okay.
And so it will look pixelated and grainy.
That was probably what happened.
So I'm wondering
keep
if
it over
that's
on
what
the website
happened.
quality.
So keep it right where it is. Compression factor of 5 is the default, but that means that it keeps it a higher quality without making it too big. So we try and balance the two. Nice resolution, but not huge. So that might have been what it was right there. Okay, thanks. Hi. Oh my gosh. How are you? Good. I'm so tired right now. I have fluid. I'm good. So this is my third of 4 sessions today. So I'm going to be tired. It's good to see you. I can't believe you finally look pregnant. You don't look enormous. You just look pregnant. Yeah, but that's normal. Before you were so petite, but now you popped.
With Mina, I never got this big. Is this free?
Great. So, I'm sorry, I'm recording.